Microplastics impair immune cells by clogging critical pathways within the body's defense system.
In this video, I'm going to be talking about the effects of microplastics on beginning with the immune cells. And so basically there are macrophages, which is a type of an immune cell. It can either be an M1 phenotype or an M2 phenotype. So what microplastics can actually do is that if they are exposed to the macrophages, there is a decreased cell viability, induced reactive oxygen species, and then triggered apoptotic gene expression. So that means that microplastics can cause programmed cell death in macrophages. Now that's not good, because this ties back to one of my older videos on adenosine. So basically cancer cells, they secrete adenosine to suppress T cells. So microplastics can suppress T cells because if the macrophages are not around, they can't engulf dying tumor cells. So what those dying tumor cells end up doing is that they secrete ATP to cancer cells so that cancer cells can secrete adenosine to suppress T cells. So reviews and recent comprehensive analysis note that microplastics may disrupt key cellular pathways in many cell types, so not just macrophages, and including oxidative stress, inflammation, DNA damage. DNA damage is one of the causes that leads to cancer. Also, inflammation drives cancer growth, or cancer migrates towards areas of inflammation caused by microplastics. Okay, so some data even shows that microplastics accumulate in cancerous tissues. Like, for example, in one study found microplastics in human colorectal cancer tissue using chemical imaging. Okay, worst is that chronic or lower dose exposure in other settings appears to promote cell survival, changes in metabolism, or even enhanced migratory or metastatic behavior due to microplastics. Now, according to some studies and some data, that there is no cure for metastasis, or at least not yet. So metastasis is basically the spreading and the growing in numbers of cancer cells. But something to know about these studies is that most of these studies on microplastics and the toxicity are done in non-cancer, non-human, or non-tumor cell lines, such as fish gills and macrophages, or use extreme doses and conditions that may not reflect realistic human environmental exposure. So many of the toxic effects associated with microplastics appear to come from plastic additives, absorbed environmental toxins such as heavy metals, or contaminants carried by microplastics, not necessarily from the plastic polymer itself. Okay, so microplastics might even affect heart blood clots. So due to urokinase, which is a plasminogen activator, and then urokinase converts plasminogen into plasmin. The roles of plasmin is fibrinolysis, which is another word for clot breakdown. Also for tissue remodeling, cell migration and invasion, which is important in cancer metastasis. So microplastics can bind proteins, possibly sequestering plasminogen or urokinase and altering their availability. So basically that means that there could be no plasmin to break down clots. Okay, so there's this other one called plasminogen activator inhibitor 1, or PAL1 for short. And basically, microplastics, they promote inflammation and may upregulate plasminogen activator inhibitor 1. What that ends up doing is that it reduces urokinase activity, therefore impairing fibrinolysis or impairing the clot breakdown. Okay, now a pathway that actually supports metastasis or is involved in metastasis is the PL3-AKT or P13-AKT pathway. So that's involved in cancer metastasis where tumors, they break out of the ECM or the extracellular matrix and then leak out into the blood, and that's how metastasis happens. Possibly after they get exosomes. Also, and the way that happens is that high levels of urokinase makes excessive or too much plasmin. And then the plasmin actually cleaves or degrades the collagen and the fibrin and fibronectin in the ECM or the extracellular matrix. And then that's how cancer cells and tumors leak out into the blood and metastasis. And to make it worse, microplastics can activate macrophages of the M1 phenotype. So M1 phenotype is actually anti-cancer and attacks cancer cells, but M1 phenotypes also produces a urokinase. So it could influence fibrinolysis or a blood clot breakdown, but urokinase also makes plasmin and then cleaves the ECM and then metastasis. Okay, so this is ongoing research, but not yet proven. But the theory is that microplastics may directly inhibit protein absorption and availability. So supposedly that microplastics, they can bind to proteins and mainly amino acids, like for example, arginine, the 9 essential amino acids that cannot be produced in the body. Uh, so basically, for example, if arginine is depleted by microplastics, then that could decrease blood flow. Also, that could weaken T cells because T cells, they need arginine to function. Tumors are actually known to steal and rob arginine away from T cells, making T cells weak, and even worse, if macrophages are secreting uh, urokinase, and then, or due to an M1 phenotype, and then the plasmin cleaves the collagen of the ECM, and then the tumors leak out, and then there's metastasis. So microplastics are having a double whammy negative effect because it's weakening the T cells by taking the arginine or binding the arginine, and then the tumor metastasis. Okay, so quick recap. Microplastics can be found in water, even food, and the air, and they are known to interact with immune cells and sometimes absorb or carry proteins such as arginine, weakening T cells. Okay, so UPA is short for urokinase, which is a plasminogen activator, converts plasminogen into plasmin. The key role is clot breakdown, or in other words, fibrinolysis. But UPA, or short for urokinase, is also one of involved in metastasis, which, according to studies, there is no cure for metastasis, at least not yet. P13-AKT or PL3-AKT is one of the pathways that is involved in and supports a metastasis. So microplastics, they boost or enable tumor metastasis in the blood. That's because microplastics promote inflammation, upregulating plasminogen activator inhibitor 1, which reduces urokinase activity and impairing fibrinolysis. Therefore, that means that if there is no urokinase, and microplastics enables or boosts M1 phenotype macrophages, which make urokinase, and then plasmin cleaves ECM, or short for extracellular matrix, and then tumor leaks out. So that's it for the video. If you want more future videos, then like, comment, share, subscribe, and keep it locked in here on my channel, and I'll see you in the next video.